Test. 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 All right. Test 1, 2, 3. Glad everyone is here today. Thank you all for coming to the first ever DeSciNYC event. Okay, it's not true. Okay, so we have a lot to cover. So we are going to, and if you're in the back, just come forward a little bit. You'll enjoy the being able to hear. And also, like a pro tip, along the edge is oftentimes you can hear people really. You can hear all of this space. You can hear really well, and like this part by the stairwell, you can hear well, but this part over here you can hear well. But you just have to go around the back. So that's sort of just like a little bit of a pro tip. If you can't hear, it's because you're in a bad spot. Okay, so we'll jump right in. So the first thing we want to start with is the DeSciNYC. So what is DeSciNYC? It's like a science meetup in New York City where you can meet and chill with other people who are also interested in science and the future and embryos and all that sort of thing. So if you're here, it's probably because you're interested in science. And there's too many run clubs and too many drinking clubs and too many bowling clubs in New York, but not enough science. So this club is the change to make that happen. So we've had so many speakers. This is actually like our 35th one doing this, so that's kind of crazy. Like there's a lot of, we've done this many, many, many times. And all the videos are all online. So the next one, we're actually gonna do one in 2 weeks. It's a surprise speaker, but mark your calendars. And the one after that is going to be on nutrient transportation and cell metabolism. So cell metabolism and the circadian rhythm are all super, super interesting and becoming more and more important as we learn about longevity and we learn about sleep and we learn about, how to be healthy. So we're trying to focus on that. And also, if you guys have ideas for speakers, come up and chat with me after, because we're always looking for people that are doing cutting edge stuff around science. What is decentralized science? A lot of people are wondering, I like science, but this is called DeSciNYC. Why is that? So science is undergoing a lot of changes right now. I don't know if you've looked at like the news, you'll see like a lot of funding is being taken out from the government. The government is no longer funding a lot of the research. And some people say this is very, very bad, but this actually creates an opportunity for science to happen at the periphery and for people to fund science on their own. And this is what decentralized science is about citizen science and allowing people to choose what types of things get funded, where their data belongs, who they want to invest in. And I think for too long, science has sort of been trapped in the ivory tower and not been able to be seen by other people. So this is like a new opportunity for a new wave of science to happen. So I'm going to chat around what types of things are going to happen with this. So the first type of thing that we can think about for decentralized science is we're going to conduct a sleep study. So we're going to look to see how sleep is impacting people. And sleep, if you guys know Brian Johnson, he says the first thing you have to do is you have to understand, you have to get your sleep in order, and then after that you have to get your that'll help you get your food in order, or your exercise in order, and then after that you sort of get your food, your sleep, exercise, food. And if you do that, you have all sorts of cognitive enhancements. You can live longer, things like that. Old slides. So sleep is really interesting, and there's two examples of people that are doing interesting sleep research in a new type of way. So, how many people here know what sleep aid is? So it's this bed that cools itself off automatically, which is very interesting. And during the OpenAI drama, when Sam Altman was ousted from OpenAI, the founder of Sleep 8 said, are people sleeping better or worse? Is there a lot of anxiety happening that's causing them to be awake more? And what he did is he was able to look at the data from all these different beds and find that people on average slept, there's a 27% increase in people getting under 5 hours of sleep during this OpenAI drama. And what's so interesting about this is this wasn't a research study that was sort of conducted in a university or that required like intense approval or anything like that. This is just something that people were automatically like giving their data, and it allowed the crowd, and it allowed a new type of person to conduct research. In a similar vein, there's this like sleep and exercise. So I've seen a lot of people here wearing a Whoop, and the Whoop is really interesting. So what this founder of Whoop did is he recently published a paper in Nature where they were looking at Whoop data and how exercise impacts sleep depending on how early or late people do the exercise. So if you exercise closer to bedtime, they found that people actually get worse sleep. But this is also another example of a type of research that is done in a different type of way than sort of in a traditional academic setting. So what we're doing is we're going to create we've created like a sleep lab that a lot of people are participating already in New York. We have over 10,000 people sleeping each night using this app that we were about to describe. And it's very cool. So it works with any wearable device. And we're trying to create like an open sleep research platform where people can contribute their sleep data and then anyone can ask questions of their sleep data as well. So we have different point systems. You can track your patterns. And we're already collecting interesting data around that. We're finding that people, when they eat closer to their bedtime, they actually like get a significantly worse score. If you're interested in trying the app out, you can download it here. If you download it and you show me after, I will give you this beautiful sleep mask so you can participate. If you're interested in sleep research, we're also hiring, so we really need some more developers and business people. We're funded by Polychain and a bunch of other VCs to try and make science distributed. So if you're interested in working with us, that is also a possibility as well. All right, so is there any questions? Okay, so, oh yeah, go ahead.
1.2 sleep point scores out of how many?
Out of 100. So it's an interesting problem of how do you like determine a score for sleep, right? So like, what is perfect sleep? Some people might be optimizing for recovery. Some people might be optimizing for longevity. Some people might be optimizing for having good mental clarity. But we basically created a normalized sleep score around REM, deep sleep, light sleep, and then created a 0 to 100 sleep score to be able to predict how well people slept. All right, so without further ado, we will go to our main speaker of the night. So human embryo editing is the future. So in the previous talks, we had a speaker who was talking about embryo selection, where they had an embryo and then were doing genetic analysis on it, and then finding sort of the healthiest embryo to implant. But this is taking it sort of a step further, where now you can actually, before implantation, you can edit the egg and select for any characteristics that you want. So this is going to be fascinating. And we have Diego from Genomic Prediction, which is a new company, which I'm sure he'll tell more about. So without further ado, let's give him a round of applause.
Thank you very much, guys. Okay. So thank you once
Let's
again.
do one picture. All right, 3, 2, 1. 3, 2, 1. All right. There we go.
Well, thank you. Good evening for the introduction. And for coming here. I know it's late at night, but this is a topic that we have been working for a while. Stefan Jadavik.
Talk really into the mic.
Here? Oh, okay, sorry.
Much better.
But really shout into it.
I had a shout here.
Speakers in the way of.
Oh, like the speaker. Can we put them down so they can see the slides?
Yeah. Yeah.
Okay. We're good there? Everyone can see? Perfect. So yes, probably you saw that tonight. Stepan Jedabek was supposed to give this talk. However, he had some issues with some data that he's going like this. Okay. So it's the first time I give a talk like that, but that's fine. So he's going to give a talk here. I think probably this summer, if not in this fall, about the continuation of this project. But today I would like to share with you a story that was a collaboration between our company, Genomic Prediction. I'll tell you a little bit of what we do, and Dieter Egli and his lab here in Columbia University. And I wanted to start this slide with this video. I don't know if you guys have seen this before, but this is a human embryo being cultured in an IVF lab. You know, we have several clinics in Manhattan, in the city, also in Jersey, around the world. And basically, this is when the first day of development, this is a time lapse of 5 days of development. Usually we culture these embryos until day 5/6, depending. Some embryos are slower and faster than others. And it is until we get the day 5, where we get a structure called the blastocyst. And this is going to be important for this talk. So I just want to put those backgrounds so that we can understand what we're doing with gene editing in the pre-implantation embryo. So these blastocysts, there are two major components. We have this bunch of cells inside, which are called the inner cell mass. They give rise to the actual fetus, to the baby, whereas that coat of cells around it would be what we call trophectoderm, and that's what gives rise to the placenta. It is important because all the testing that we do is from the trophectoderm, okay? So we don't touch the actual cells that become the actual fetus. Now, again, I want to give you the setting of where this magic happens, and that is in the embryology lab. So the embryology lab, they are different from clinic to clinic, but basically, this is what one looks like. This is one in Panama, one of the best of quality in the world. And you will see this micromanipulators with microscope. All this imaging that I'm going to show is happening in a petri dish, right? So sometimes it's just hard to visualize how this is taken. So imagine that all this is happening here with what we call a micromanipulator. All those needles that you will be seeing in the videos is like a joystick. You play with it. And then you have a pedal to move it. But these are things that sometimes are not shown, and I just wanted to clarify the setting of where this magic happens. Now, before we start, I do want to clarify two different concepts. One is genetic selection, and one is gene editing, which is the topic of today's talk. The reason why I say it is because sometimes I hear these terms interchangeable, and they are completely different. Genetic selection is something that we're doing every single day in IVF clinics that do PGT, or genetic testing. Now, before we go to gene editing, I did want to go over briefly what genetic testing means. You're going to listen to me saying PGT a lot. So PGT would be pre-implantation genetic testing. That's what our company, Genomic Prediction, do. So in pre-implantation genetic testing, of course, you need the embryo. So these embryos comes from the clinic. Again, you put the egg with the sperm, fertilize it, let it culture. And then once you have that blastocyst, that embryo that I showed you at the beginning, then you're going to take a biopsy. I'm going to show you a video shortly about how that looks. That biopsy is sent to the lab. We amplify the DNA, we interpret the data, and then we just give a PDF to the clinic and the patient so that they can select which embryo would be transferred into the womb. Now, this is a video of a biopsy, again, just for you guys to see how this happens. We have this embryo. The shell is called zona pellucida. Some cells are coming out. We take that biopsy from those cells coming out that would eventually, remember, become the placenta. And with a laser, it's like a pedal, so it's actually like a nice instrument. And once you do the laser, you kind of suck it, then those cells are put in a PCR tube and sent to the laboratories. What happens to this embryo? It goes to liquid nitrogen. And it could be there for years, right? We have pregnancies, I think, after 20 years of an embryo being biopsied and tested. So nothing, like we were talking before about turnaround times of genetic testing. In liquid nitrogen, that's not really an issue. Okay, now that we clarify that concept, gene editing is completely different. Because in gene editing, you're actually messing with the sequence of DNA. So you're making changes. Again, genetic selection, you're just screening, seeing what the embryo already has. With gene editing, we are actually changing the DNA of those cells. Now, you might have heard the term CRISPR-Cas9. For gene editing, there are several tools. This is not the only one. However, this is the one that's been more developed. There was a Nobel Prize to two people who developed this. CRISPR-Cas9, I won't get into detail, but it's a system that already bacteria has to cope against, foreign invasions of virus or other kind of organisms. But basically, the CRISPR-Cas9 became so useful because once you understand the components and you get them into the cell, it's actually pretty effective in doing this job. So CRISPR-Cas9 has four major components. One component would be the Cas9. The Cas9 protein is this Mickey here. So it's an enzyme. It's a protein that goes to the DNA and cuts it. That's component number one. The second component is what we call the guide RNA. So the guide RNA is a molecule that you can actually design the sequence whatever you want. There is a company called IDT. So you send them the sequence, what nucleotides you want, and they will send you a tube with some powder, which is lyophilized primer. You put some water, you have it there. It's very important because then you can design whatever sequence. You can target it to any location in the genome. The third component is, of course, the target DNA. And in our lecture, we're going to focus on the DNA inside those embryo cells. And the fourth component is something that gets ignored when we talk about CRISPR-Cas9, but it's extremely important. And I try to show it here, which is the cell DNA repair mechanisms. Because without this, CRISPR-Cas9 is just useless. So I'm just going to take it out because for purpose of showing how this works. So how does gene editing work? First of all, we have, and I won't go in detail into that, but there is a whole research on how do we get these components into the cell. You could get them, of course, by injection, which is very easy, but it's very hard to inject, liquid into a single cell, which is more than microscopic. You could do electroporation, many other techniques. But let's just pretend we're already inside the cell. So we design a guide RNA to a specific sequence, and what will happen is that guide RNA would hybridize, would match to the DNA. So it would kind of open it. This is happening in the cell. It puts the whole machinery there, and the cut happens. It's literally a cut. So it generates what's called the double-strand DNA break. And this is not editing. This is just cutting the DNA. The editing happens when the cell DNA repair mechanisms go and fix this. Because when that's fixed, it's not perfect. You introduce novel nucleotides, insertions, or you can delete nucleotides, deletions. Or you can actually, let's say if there was a T in that position, the cell could just put an A. So that is how the gene editing actually happens. It's not that you are going and customize and put an A there. You are relying on the cell repair mechanisms, and therefore, as of now, it's somewhat random. There are some technologies that are trying to be more precise, but this is the concept of what happens. Now, in 2017/18, there was a major publication of our field from the group of Dr. Mita Lipov in Oregon. And this was the first proof of concept in the human embryo of a successful gene editing. So basically what they did, they took the egg, they fertilized it with a sperm, then they let it culture one day. This is the stage of a zygote. You have the two genomes, mom and dad. And at that stage, they injected the cocktail of proteins, the components that I showed you before. Now I'm going to show you a video that has nothing to do with this paper. It's from our laboratory, but just so you know how these injections happen. So basically you have it there, you load your needle, you put it into the embryo. That's how it's happening, okay? And then you let the embryo culture, right? This embryo was cultured until day 3 of development, when you have around 8 cells. We call them blastomeres, and then you analyze them. So the experimental design was pretty elegant in this study, because what they did was to find a sperm from a human who was already affected with a recessive mutation. So all the DNA in that sperm had a mutation, right? Therefore, the only outcome in that embryo was to be heterozygote, meaning that it will have the mutation, for sure, because the sperm already had it. Meaning that if this actually was working, then we would expect some cells to be fixed or not to have that mutation. So this is what they did, exactly like that. Mutant sperm, put it in the egg, and then they got several embryos, of course. I don't remember how many, but one of the outcomes was what was expected, which is all those cells would have the mutation. So their CRISPR-Cas9 did not work. But then in some cells, they started seeing that some cells actually didn't have the mutation at all. And most importantly, few of those embryos, not many, because you can see it's a mixture of things. Again, it's very random. But in some of those embryos, all the cells did not have the mutation. So you would understand the impact that this had in our field, because for the first time, we were fixing or curing a mutation in the embryo before it's transferred into the womb. The problem here, and what happens in science, and something that I have learned in my years of this field, is that you have to be very careful over interpreting data. The analysis that was done in those cells, those blastomeres, those embryo cells, was pretty shallow analysis. And just because you didn't see the mutation, the assumption was that you cured it, right? It was also in the news. And Dieter Egli, which is Stefan Jarabek's mentor and with whom we collaborate, started writing these papers trying to provide different interpretations to the same finding. So why? Hold on, maybe you are not curing the mutation, maybe you're just not seeing it. Maybe that chromosome was not even there. And of course, unfortunately in science, there is a lot of egos, right? And then you start seeing all this back and forth, back and forth. So even the authors of that initial paper, they brought up this article saying that this was, yeah, it made a lot of sense, but was sheer speculation. And it was true. It was just speculation. And if you want to prove a concept, you just have to get it done. So what we decided to do with Dieter and Brixton was to replicate the experiment, at reproducibility. Because reproducibility is something that it's essential in science, especially when you're doing a biotech company or something like that. You need to have this reproduced in a different setting by different people with different interests. So basically, we follow a very similar experimental design. We took a sperm from a guy who was blind, so he had both of his copies of that gene had the mutation, they were affected. The gene was EYS, so the guy was blind, he donated the sperm. We took donated eggs and we fertilized those eggs with the sperm from the blind guy. And then the experiment was to, once we got the embryos, inject the CRISPR-Cas9, direct it to that EYS gene, make blastocysts, the day 5 embryo development. At this point, do PGT, which is taking some cells from the trophectoderm to make sure that the editing happened, right? So that was the first proof of concept. And another thing we wanted to do, and we're still doing, is because this embryo is not going to get transferred. We can talk about that, like in the future, when are we going to transfer editing embryos? But these embryos, we took the inner cell mass, which develops into the fetus, and developed a embryonic stem cell. Because when you develop an embryonic stem cell, you don't have 100 cells to play with. You have hundreds of thousands, millions of cells. And this is important because what if this CRISPR-Cas9 is cutting somewhere else? We are directing it to cut to one gene, but it could certainly cut somewhere else, and that's what we call off-target effects. And until we have safety data, I do not believe it will be a transferable embryo into a patient. So anyway, now, one thing that was a game changer for this study, remember that I told you before that there was some suspicion that there was overinterpretation of data. So how do you analyze that cell to make sure that you are actually not seeing that mutation and you are not causing any other harm? There are several ways to analyze embryos. And this is another talk. Several technologies. You could do FISH, you could do something called array genome hybridization, you could do next generation sequencing, but there is a specific platform called SNP-array. So a SNP-array is a platform that we use for genomic prediction, and it genotypes up to 800,000 locations in the genome, And these SNPs are small locations that are polymorphic. So maybe some of you would have an A allele or have a B allele. So they're highly polymorphic. In fact, you can actually predict ancestry based on just these locations. So anyway, long story short, we had access to this platform and we just decided to use it on those cells just to make sure whether the editing was happening correctly or not. Now, before I show you the results, I do want to walk you through how we read this data. So the company that we work at, it does PGT, so it's a commercial company. We processed last year 40,000 embryos, and this is a small company. Like, these are embryos being processed every year. So the plot that we get, it's called the violin plot. So remember that we have 23 pairs of chromosomes, well, 22, and then the sexual chromosomes. So here, each chromosome is represented by one of these diamonds. And what this means, basically, I don't want to go in detail, but as you could see, the y-axis says allele frequency. So if we have 2 copies of each chromosome, we would expect the vast majority of SNPs of these locations that are heterozygote, meaning differing alleles from mom and dad, to be clustered around 50-50, because we have 2 copies of the chromosome. So that's why this diamond kind of localizes to 0.5, because this is confirming that this embryo has 2 copies for each specific chromosome, except for chromosome 16. Again, this is just an example. For chromosome 16, you can see that there is no data in the middle. And what this suggests is something in genetics that we call loss of heterozygosity. That is because there's only 1 copy of chromosome 16. And this is therefore an abnormal embryo. In fact, we have so much resolution per embryo. Remember, this is coming from just 6 cells that are being tested. You can visualize the entire chromosome, short arm and long arm, and you can see that there is no data in the middle. So this is how we do confirmations that there is a loss of a chromosome. And of course, the report of that patient would say, you have an abnormal embryo with a loss of chromosome 16. Don't even waste money and time transferring it, because it will result into an abortion or, a biochemical pregnancy. Anyway, the reason why I show you all this is because this tool was not used before in the previous research. And as you could see, it gives you a lot of information on these cells. So now I'm going to show you the results. So just to recap the experimental design, we took the egg that was donated, the sperm from the guy who was blind, fertilized the embryo, injected the CRISPR-Cas9 cocktail, developed the embryo until day 3, and then until day 5, we took the biopsy for PGT and tested. Remember that gene that we had for this specific experiment was in chromosome 6. The gene was called EYS, and is located here, close to the centromere, which is that part that unites both short and long arm of the chromosome. And this is one example that came in the paper. So as you could see, this is, for those who work in the IVF industry, this is a very beautiful embryo. It's very good morphology. So that was good, because we were not messing up with the morphology of embryo development. And once we visualized all the chromosomes, but this is just the example of chromosome 6, short arm, long arm, we started seeing something that was unexpected. If you look at the short arm, remember what I showed you before? Here you see data around 0.5, which is expected because you expect 2 copies of each chromosome. But what was very unusual is that right at the point of cut of CRISPR-Cas9, we started seeing loss of heterozygosity. We thought at the beginning that it was a de novo, what we call deletion, because this means that you only have 1 copy of that part of the chromosome. Because it happens every day, like you see de novo duplications or deletions, but it kept happening, and always right at the point of cut that we designed the CRISPR-Cas9 to go. In other embryos, the finding was even more striking. We were seeing loss of heterozygosity everywhere in the chromosome. And after doing so much data and seeing the same thing, we concluded that CRISPR-Cas9, unlike what they interpreted in the previous paper, it's not that it was getting rid of the mutation. It was getting rid of the entire chromosome, or part of the chromosome. So that's why CRISPR-Cas9 was actually not fixing the mutation. It was eliminating the entire chromosome or part of it. So you can imagine the implications of this. You know, this was an unexpected finding. We presented in several conferences. There was a lot of, I think I have a couple, well, this is the paper that came out from Michael Zuccaro. It actually helped us to understand a lot of cell repair mechanisms in the embryo, but the major finding was that CRISPR-Cas9 was not doing what it was intended to do. It was actually eliminating DNA. Now, the cool thing, again, I always emphasize on reproducibility. After some groups saw these findings, they replicated the Francis Crick Institute in London, which has a lot of prestige. Also, in the European meeting, there is also other groups using the SNP array, and they're finding exactly the same. Now, again, you would expect when things go okay, people are like, okay. When things go wrong, everyone is on you, right? So if you could see all these titles of all these major newspapers, CRISPR is bad. CRISPR is not ready. CRISPR will never be the thing, the solution for gene editing. And it's true, because we found something that wasn't expected. And remember, we need safety data before we do this in the embryo. But there is something in the field of infertility that we have to cope every single day, and that is embryonic aneuploidy. So embryonic aneuploidy, first of all, is the most common genetic abnormality in humans. What does aneuploidy mean? Remember that we as humans, we have 46 chromosomes, 22 pairs plus the sex chromosome. If a human or an embryo has more chromosomes than 46 or less than 46, that's considered an aneuploid. And what's even worse, at least for our field, is that aneuploidy is the most common genetic abnormality of infertility. So this is a plot that we did when I was training in RMA of New Jersey, a huge IVF program in Jersey. So we took patients from all maternal age, and as you could see, the older the woman gets, or the egg gets, the higher the rate of embryonic aneuploidy. Because remember that women are born with all the eggs that are going to be produced during her lifetime, right? So a woman who is 40 or 45 years old has eggs that have been there since she was born. So the aging of eggs results in having embryos with aneuploidies, more extra or less chromosomes. But the reason why I bring this here is because for infertility, this is a huge issue. That's why in infertility clinics, we get a lot of older women, because they try and try and try, and the reason why they don't get pregnant, most of the cases, is because their embryos have an extra chromosome, for example. So it was actually sitting down and be, okay, we have potentially a solution to eliminate chromosomes, which are already a headache for the infertility clinics. And that was something that we started doing. So again, I won't get into detail, but this was the first time that we could find a tool that could get rid of embryonic aneuploidy. So cure trisomies, for example, cure Down syndrome from the preimplantation embryo. So the experimental design, basically, it was to take the egg. Remember that an egg is produced through a process called gametogenesis, and then you have a stem cell that divides into two cells, and then these two cells into other two. And there are two cells that are a byproduct of this division, which are called the polar bodies, right? And the polar bodies are these small cells that get DNA because it's a division, so they get equal part of DNA, but they just don't get the cytoplasm and the rest of chemicals. Only one cell becomes the egg. But the reason why it becomes more important is if we can actually analyze the polar body, and let's say one polar body has an extra chromosome 16, we can assume that the egg has the missing chromosome 16, or vice versa. If the polar body has a missing chromosome 16, we can assume that the egg has the extra chromosome 16. So the experimental design was to do this, and then if we find that the polar body has a missing chromosome 16, why don't we inject CRISPR-Cas9 directed to that chromosome 16 so we can get rid of it? And this was actually performed here in Columbia University. So Jenna Turosi, who was the fellow by then, she started doing these experiments again. You will analyze the polar body. Actually, I show you the biopsy of the embryo, but back in the day, before we didn't know whether we could touch the embryo, we were doing polar body biopsies, right? That was the gold standard before. Now no one does it because the egg is extremely delicate and most of them die. But in this case, we managed, I mean, it's work for research. these are all human eggs. We took the polar body, we analyzed it, and some of those polar bodies, at least for this egg, one has a missing chromosome 13, but the other one was very useful because it didn't have any signal of chromosome 16. So we could assume that the extra chromosome was in the egg, and if that egg was going to get fertilized, that would be an embryo with 3 copies of chromosome 16, the 2 maternal and the paternal. So after we did that, we designed a guide RNA. Remember, this is the part of the CRISPR-Cas9 you can design, directed to the chromosome 16 from the mom, because you didn't want to get rid of the paternal chromosome 16. It's important that you have the 2 copies of each chromosome, one from mom, one from dad. Not in all, but in most chromosomes. So anyway, what happened is that we did this injection, we cultured this embryo until day 3, we took each cell and analyzed it, and we got actually a myriad of results. Some of them had 3 copies of chromosome 16, so we confirmed that our assumption was correct. Some cells had that extra chromosome. Some of them duplications, deletions, right at that point of cut that we designed the CRISPR-Cas9. But what became more striking and surprising was that some of those cells have only 2 copies of chromosome 16. And this was the first time that has been reported that aneuploidy correction can happen in the human embryo. Now, this of course opened the door to many interviews, many things. It was actually awarded a prize for the ASRM. The ASRM is our society in the US. It's called the American Society for Reproductive Medicine. Genaturously got the first award because this was something extremely unique that no one else had reported before. Now, of course, we're very far away from applying this clinically, but the research continues. And this is already, we have some reviews, I think, in Nature Communications, but right now it's in bioRxiv, if you are more interested in seeing the detail and the minutiae of this project. And, then the press started to get more positive. So, okay, CRISPR is not that bad. Maybe CRISPR could have another application. In fact, people are trying to get into it, and there is a lot of other reports, not in human embryos, but they're doing in cell lines. And it's these guys are right. This is possible. So again, reproducibility, and hopefully in the future, we could have some solution to do this for people who are 45 years old, their eggs are all abnormal, and this is the only chance to have a baby without recurring to gamete donation. Okay? Now, this is just, I'm almost done. This is just another, let's say, application of this. It's specific for very, very rare cases, but we all have the chance, or should have the chance to have a baby of our own, genetically. So there is something called a Robertsonian translocation. So this is just a patient, a karyotype that we generate. When you want a karyotype, you just want to know how many chromosomes you have. And for chromosome 21, there are some individuals that do have 2 copies of chromosome 21. However, they are attached to each other. That's what we call the Robertsonian translocation. Why is this a problem? Those people are normal, phenotypically. The problem is that when they make sperm, or when they make eggs, then they are going to have half of those sperm and eggs with null disomy 21, meaning no chromosome 21 because the 2 copies are attached. And the other half of the sperm or the eggs are going to be having 2 copies because they are attached. So if you fertilize an egg with that sperm, it's going to be Down syndrome, no matter what, or vice versa. So imagine if we could have something like this. We have this guy with this problem, that condition, again, phenotypically normal. We get an egg, we get this embryo, but what if we can get rid of that extra maternal DNA? Now, of course, the baby would have the same Robertsonian translocation, and we can discuss ethics about it, but at least this could be a solution for that person, that couple, to have a baby that would be phenotypically healthy. Okay, so this is basically what I had for you guys. This is a story that continues. We started this collaboration in 2021. Now, there are so many questions that have arisen after this project. For example, when you inject this CRISPR-Cas9 cocktail, it remains there. You cannot take it out. So how do you make sure that it stops working? You don't want it to keep editing. So Alex Robles, he also presented in ASRM 2022 we were trying to do a second injection, which is an inhibitor of CRISPR-Cas9. So we could stop, tell him, okay, do your job at the beginning when you are one cell, but stop when you are 2 or 3 more cells, because you don't want to have mixtures, right, of these karyotypes. So this is something that's already in bioRxiv, also under review. Ivan Rashev is also a fellow from Columbia University, and then we went to another chromosome which is problematic, which is chromosome 18, also presented at ASRM 2023. Last year we presented some data that is very enticing and very cool, because I'm talking about eliminating chromosomes and aneuploidy. But remember that initially we wanted to cure a mutation, right? And that's a project that keeps going, and hopefully Stepan Jedabek will tell you a lot of good data of how we found a solution to do this accurately. Okay? So, and I just wanted to end this talk with this quote. Nathan Trevis is my mentor, also the co-founder where I worked. And I really like it because I do agree with it. I don't know if you can read it and maybe discuss about it. But the future of IVF, gene editing, and preimplantation genetics is precisely curing disease before pregnancy. This is what we should aim, what we should go after it. We were talking before about companies offering IQ testing or skin color, cosmetic traits. We can debate that, but at least curing disease before pregnancy should be the way to go. And with this, I of course want to thank DeSciNYC New York, but most importantly, well, of course, my lab, my team. We have a lab in Hackettstown, New Jersey. And most importantly, where the magic really happened, which was Dieter Eggle's laboratory. This is when they came to Genomic Prediction to pay us a visit. This is Stepan, that he will come probably later, Marcus Kuhlmann, and of course, Columbia University. So here is my contact, and hopefully you enjoyed the talk. Thank you.
Does anyone have any questions? Yeah, go ahead. Do you want to come up, and then his is being live streamed also. Just here, I'll come all the way. Okay. Just take the mic. Okay.
Yeah, you mentioned this like goal at the end, like curing all these disorders. Do you think people who are thinking about having kids should like wait? Is this like an imminent development? Are you confident in your timelines at all?
So when it comes to gene editing per se, I don't think we should wait, because it will take time until this will be available to transfer an embryo. However, there are other things that are available that is not editing. For example, the embryo selection. So for that, that's available. But for gene editing, maybe I think 5 years will be, underestimation for gene editing. But for example, selection of polygenic traits or avoid some mutation like cystic fibrosis, that's something that is being done nowadays.
Yeah, come up and take the mic.
So if I understand correctly, CRISPR-Cas9 doesn't do precise editing, but it can knock off entire chromosomes. And I think in your research you showed that you were able to use CRISPR to knock off the chromosome, but not all the cells get the chromosome knocked out, right? So in that case, is it ready for implantation, or do you still have work to do because not all the cells have the same antibody?
Yes, of course. There is a lot of work to do because we just tried it in chromosome, I think 18 and 6, and some of target effects, we don't see it everywhere in other chromosomes, but we have to try them on all of them, right? So that was one, and then you mentioned something at the beginning that I was going to answer it.
It didn't work for all of them. Right?
Oh yes, so exactly. So again, when do you inject it? Because if you inject the CRISPR-Cas9 cocktail after you already injected the sperm, you're going to have something called mosaicism, which is like it's very hard to control. But what we're trying to do is injecting this cocktail at the time you inject the sperm. So when you inject the sperm, you kind of load it in that micromanipulator, and you can put the needle on like a drop with all these cocktails, it sucks it. And if you inject it at that time, then you have less chance of having mosaicism or different cells with different, karyotypes. But that's something that we're still researching.
Oh, yeah, here.
Oh,
yeah.
sorry.
I wanted to ask you about the scientist in China that years ago CRISPR basically corrected or introduced a mutation in twins, human twins, to make them resistant to HIV infection. The twins were born and they were, they're growing up now. So I was wondering if that was luck or they were very good in selecting the embryos, making sure that the procedure didn't delete chromosomes.
Exactly. So, probably some of you have heard about that Chinese scientist who did this some years ago. And he claimed in a conference that he did this CRISPR-Cas9, transferred the embryos, and indeed, these babies were born, resistance against HIV. The problem with this is the secrecy behind it, right? So there is even the Chinese government got involved. So this guy was in jail, and he's no longer in jail. He's actually having his research lab. So the secrecy behind it has kind of blinded us to know exactly what's happened there. The babies, and I think the couple are also like under some kind of protection. But this is something that was such a huge hit for the scientific community that they got kind of isolated, after he went in jail. And unfortunately, we don't have those reports. And this is very important because what I'm showing you here and what you will read is what's being reported in literature, right? There is, imagine all these crazy people in like these isolated islands. You don't know what they're doing. We were talking before about Mexico. In Mexico, there are all these laboratories doing all sorts of things, right, that we don't know. But to answer your question, there is a lot of secrecy behind it, but the guy claims to do it. It's just that we cannot even sequence it. I wish to sequence the genome to see what we find.
I would say it's probable that if they didn't result in live births for that type of thing.
What?
No one, the likelihood that resulted in like a live birth, I think, is near zero. Hui.
Yeah, right. That's
Yeah.
what he claims.
Yeah. So it's like the guy who claims he invented Bitcoin, but then like doesn't back it up with any proof. Is like it's probably just like riding off of like the hope that it could be true, but I don't think it actually worked.
All right.
Yeah, really interesting stuff. So for the aneuploidy correction, this is happening before it's an embryo, right? First off? Okay. And what I was wondering was, do we know why the CRISPR-Cas9 takes out an entire chromosome rather than replaces a part of it, or and then also does it only take out one of them and leave the other when you do it in an aneuploidy?
So, regarding the first question, so we do these injections once you already have an embryo because you already have the sperm inside. So that the embryo with the two circles, that means it's already what we call a zygote or embryo day one. And we want to do it there because if you go beyond, then you're gonna have mixtures. But yeah, when you have the day one of an embryo, that's number one. Now, why is CRISPR-Cas9 cutting and getting rid of all these embryos? Of the chromosomes. There are several reasons for this. What we found is that, remember, like I showed you, you have a chromosome and you have what's called the centromere, which is that part that unites both arms. We found that the closest you cut to the centromere, the higher the rate of eliminating chromosomes. And that is because these chromosomes, they use the centromere to attach what's called the spindle. The spindle is some sort of a bridge that the chromosomes locate there to separate for cell division. So what we believe is happening is that once you cut there, maybe the DNA mechanism is not perfect. Actually, we found out that it's very not perfect in the human embryo. And because of this lack of repair, we believe that that little cut could cause the whole loss of that chromosome, right? So, and there was a third one that I forgot.
So in an aneuploidy, does it take
Oh,
out
yes.
copies?
So, remember that when we wanted to cure the aneuploidy or get rid of that extra chromosome, we needed to be very careful because we didn't want to get rid of the two normal ones, or we didn't want to get rid of the paternal. That's why of all these embryos and, sperm and eggs that we have for this research, we also got DNA from the gamete provider, from the guy or the woman. Because then you have to sequence it, so you can be, okay, this chromosome has this SNPs or this sequence, which is specific from the egg, and this is the one we want to cut. So we designed a guide RNA specific for that extra chromosome that was from the mom. Exactly, because you don't want to get rid of the paternal one or the other normal one.
Why don't you remove the chromosomes at the egg stage? Why?
Because at the egg stage, in order to know whether there is an extra chromosome or a missing chromosome, you have to have some sort of information. So that's why you have to do the polar body, right? And the polar body gets extruded once you get the sperm inside, right? So once you get, actually an egg, it's so cool because the egg hasn't actually done meiosis or become an egg until the sperm gets in. Once the sperm gets in, there's a phenomenon called egg activation, and then it extrudes that second polar body, and it becomes an actual egg. However, at that time, it's already an embryo.
Do you still get the chromosomal deletion from single-stranded breaks, like using prime editing, for example?
So, I won't answer that because that's Stepan Jerebek's talk. So, yes, remember something very important. CRISPR-Cas9 is a huge, let's say, I always imagine there is an umbrella, you have CRISPR-Cas9, and you have all these different systems. CRISPR-Cas9 base editing, CRISPR-Cas9 double-strand breaks, or there are a myriad of CRISPR-Cas9. The one that I'm presented here is the more canonical version that cuts both strands of DNA, but if you manage to cut maybe one strand, maybe you won't see that result. But hopefully that will be, the product for Stepan's talk.
What are the ethical concerns of this science, and where do you see it converging?
So ethics is very hard to give an answer because what I've learned is that ethics is a dynamic concept. It depends on the society. I'm from Costa Rica originally. I can tell you that the ethics behind this is very different from the US and even Europe. I mean, what I could answer is mostly based on safety. I believe that if we were to do this study here, we will need a lot of data or safety data until we can actually transfer it. But ethics is something that we all will have our own opinion, and depending on our background also.
What diseases can you practically cure today?
Say it, Ben. Say it into the mic.
What diseases can you cure today?
Okay, if you mean cure with actual editing, if you think of it, any disease that has a single mutation, because for example, we have several things. Let's say cystic fibrosis, that's a small mutation, one letter in the nucleus, in the sequence, that will be easy to do. In fact, we have some embryos that have been cured. I don't want to use that term, but that's something. Fragile X and Huntington disease, these are repeats, expansions. That's something that we don't even start thinking of how to do it. So I would think that everything that's a single mutation could potentially be fixed. But again, potentially, because another assumption we make is that the whole genome is the same, and there are some places in the genome that are so hard to get in, so hard to cut for some specific epigenetic things. But as of now, I would say if it's a specific mutation, that's pretty possible to cure or to edit.
Do you have examples?
Cystic fibrosis is one, for example. Beta thalassemia, I believe, it's another one. I mean, there is a myriad of these diseases, but point mutations, I would say.
What are your predictions for the next 5 years of, advancements you expect in this field?
So in the gene editing field, I think the predictions is we're going to have more off-target effects analysis. Remember what we do in these embryos, we don't throw them away, we create embryonic stem cell lines, because we have to make sure that it's not damaging other parts. So I think that's going to be a huge area of research. Maybe AI would give us a tool to try to cope with this, because it's just a lot of data and a lot of positions. So that's something that I actually do not think there is a biotech company or a niche to explore this, but this is for sure something that will be pivotal before we actually transfer an edited embryo.
How far do you think you can get with the technology that we currently have? We currently have CRISPR-Cas9. there's certain things we can do with it, and not all of them are research. Like, how far can we go with that, so how far can we go with that, and what is like we need something else?
So when you say how far, you mean in terms of what we could do in the genome? Or?
Yeah, what you can cure, we can do.
Well, I mean, how far? I was a fan of Jurassic Park, when I was growing up. And it sounds silly, but there is a company called Colossal in Texas doing things very similar to what they're doing. Listen, the genome is something that, as long as you understand the raw material, it's something that it's very easy to do. Like all these sequences that you hear, you just send them you do an in silico model, you send the order to the lab, and next day you have the primer. So how far? It's hard to say, but I mean, you could edit, I don't know, one thing, for example, people ask me like, cure disease, whatever. What else can you do? There is something called protective variants. There are some humans, for example, that have mutations that don't cause disease, but could be protective against Alzheimer. So imagine someone who has family history of Alzheimer, and you can use gene editing to actually not fix a mutation, but introduce a mutation that's protective against Alzheimer. So I think that's something that not many people have discussed, but to my knowledge, I think it has a lot of potential.
This process shows.
Here, you want to come up, so we can hear.
So my question is, how would you edit a genome for a human that's already alive, as opposed to editing like the origin cell of an embryo and then having that propagate?
Yeah, so this is there is a whole area of research of gene therapies using CRISPR-Cas9. Now, of course, this is another scope, because here the point is just to have all cells in our body with the same editing. But for example, I think for sickle cell anemia, what some groups are doing is that they extract cells from someone who is affected with some disease. They use CRISPR just to fix that population of cells, to make them, let's say, wild type or not have the sickle cell mutation. You re-inject them, or you put them back into the blood of that person, and then I think there are very good results. So in terms of editing the adult, I think it will be very much localized to a specific type of tissue or specific type of cells, but this is happening, and these are the gene therapies for sickle cell, for example.
So when people are thinking about doing in vivo gene editing, it's like the edit on an adult is like obviously like mosaic, right? In fact, you want it to be mosaic because you want to target like specific tissue types and stuff. Like being mosaic is considered a success, right? Like you want to edit every cell. And so like given that we're so like fine with having like mosaic adults, why is mosaicism considered so like dangerous and something to avoid in human germline editing?
I think the major reason for that is that those cells at that early stage, there are different stages, but they are called pluripotent, multipotent. So for example, one of those, that inner cell mass, which is what becomes the fetus, it's just like 100 cells. And that would develop into all the tissues and organs that we have. So I think the reason why mosaicism is such a problem in the embryo, on the early embryo, is because you don't know what's the destiny of that cell. What if it's going to be a brain? What if it's going to be just the skin? So it's because at that time, it has the potential to develop every tissue in the body, and that's why it becomes so hard, because you don't know the fate of it.
Yeah.
One thing I didn't understand was there is like a 1 in 4 chance it successfully works in the Rust Bell when you do the CRISPR editing.
So yeah, this is certainly not 100%, because remember, we are relying on the cell repair, DNA cell repair mechanisms. So part of Stefan's talk is like some of the efficiency we have got with this editing, around 80%. But as of now, we don't get there. And the reason is mostly because it's some of an stochastic approach. We depend on what the cell is doing. So maybe there is another niche to do some research. What does the cell need to do its job well, right? And that's something that we haven't focused. But I believe that's something that could justify how inefficient it is as of now.
So what's the probability now that it works?
This depends a lot on different settings. At least in our project with Stepan, we have around 80% editing, successful editing on all cells, but 80% is still something that would be clinically not acceptable, right? But we'll have to get maybe 99 and up.
The very first paper was like 10%, right?
The metalipop, yeah, but again, it was not actual editing. Then we discover a misinterpretation. But yeah, it depends also on the CRISPR-Cas9. Also, we have
Oh,
one
here,
that...
go for her first. Yeah.
No one...
So you kind of briefly mentioned that some of your competitors are focusing more on these more controversial behavioral traits, IQ, that sort of thing. Do you think that that's like leaping ahead too early in the field and the field really needs to focus on disease right now, or is that interesting science to explore whether or not these polygenic risk scores for behavioral traits have any value?
Yeah. So this is I will answer it. It's just nothing to do with gene editing. This is embryo selection. So there are some companies who are doing this PGT testing and trying not only to predict risk of disease, but also predict the IQ of that embryo or cosmetic traits. Now, my opinion, again, this is my opinion. I believe that as of now, we should focus on clinically relevant disease because, you know, these are patients after all, right? And we don't want to do harm. But then you start with the ethical question of reproductive autonomy. This is something that in the US is you know, liberty, right? Something very important. In Europe is something that not even think about this. So that's why when it comes to ethics, this depends on the context. We get a lot of patients from East Asia and they just want the perfect baby. You know, they want the most intelligent. So it's something that, yeah, in our company we don't offer, but we get requests all over the place. Is it right or wrong? I'm not in the place to answer it, but it's certainly possible. That I can assure you. It's certainly possible and technically accurate also.
Did anyone want to ask the question yet? Want to ask one?
Oh.
All right, yeah.
So I'm just wondering, rather than going through the extremes of gene editing or deletion in CRISPR, is there a way to more, I guess, naturally stimulate gene expression, whether in you know, younger chromosomes that are just expressing themselves or adults to kind of optimize how they express themselves? Because, you know, we all have same 23 chromosomes. It's just how they are expressed that we get certain diseases or certain benefits from it.
Well, yes. So the question is very interesting because it's without, you know, messing with everything. Like, is there some sort of environment maybe that we can create that would decrease the level of the disease? This is something that in our field of the IVF industry has been, of course, a holy grail. Like, we all want to go there. We want minimal invasive procedures. The thing is that as of now, nothing has worked. So the pre-implantation embryo is delicate, but it's very strong. Like, those blastocysts, you know, you can make them in an incubator even with mold. Like, they are pretty strong. Sometimes we believe that, we were talking about that before, like nature is perfect. I disagree. I think we as human beings, Homo sapiens, we need to understand nature, but there is nothing wrong with going beyond it or trying to perfect it, at least to avoid disease in the embryos. But to answer your question, there is a lot of approaches, incubators with different gases, with different environments, but as of now, nothing that you can see an actual cause or effect.
One more. Yeah. Go ahead.
For the aneuploidy correction, what is the roadmap for that? What's blocking you from making that viable?
Funding, well, several things. Funding, I mean, of course, it's an idea that we have, of course, presented it several times. There is a patent behind it. I think part of the issue is that it's a very complex thing to implement. It sounds pretty cool, but you have to do a polar body biopsy, and you have to go through that. Nowadays, women who are 42 years old, they still make embryos that are euploid, which means no aneuploidy. And we already have a screening tool. So we already have a screening tool that is $200 per embryo. So this would be specific for specific cases, and I think that's one of the pushbacks, but the research is there and there is a lot of people interested. One more.
How's it going? So the question is, I guess, if this can be responsible for like increasing IQ, going back to that, will it have like cascading effects down the line? See like 300 IQ or something?
So with this IQ, it's first of all, it's the heritability of IQ, meaning like the background, the genetic background is pretty strong. That's why it's so accurate in prediction. Now remember that when you talk about selecting for IQ, you are doing a screening. It's not that you're editing and making the 300 IQ. You are just selecting from what you have. Actually, we get sometimes questions like, we want like this high IQ. Well, first of all, we don't offer it. Second, we can only select from what you're offering, right? That's true. Sometimes you want the tallest, and we are sure like that cannot be done. But I would say that, I mean, a lot of people are interested here. You have no idea the amount of emails that we get from all over the world. Do we do this? Do we do this? Is there going to be potential gene editing to increase IQ in the future? Listen, is it technically possible? I don't see why not. But is it going to be implemented? Then that's another question. But it's mostly screening right now, right, of a pool of embryos that you generated.
All right,
That I don't know.
One more question here, and then we'll do personal. He'll be around after.
What can we do like day to day, like in our daily lives to get like better sperm, better embryo, like ourselves without gene editing?
So to improve the quality of gametes, there is a whole industry of this. You know, I think that was your question came. I mean, that's not my expertise, but certainly if you follow our industry, the IVF, there's a bunch of companies trying to, what can you do to improve the quality of the sperm and eggs? We were talking about egg freezing before. They asked me for my advice. I think beyond 35, that should be, and you don't have kids that you would like in the future, to freeze your eggs. With the sperm, after 40, I think that would be also a good thing to do. But that's another industry that I don't really cover.
Two more. Okay, go ahead.
We were talking about this a little before, but what's like the best way to mitigate against like potential unintended consequences of like optimizing for certain genes, and like what's out there right now?
Yeah, so we were discussing at the beginning of this talk that there is a very legitimate criticism against selecting for these things. Because what if you are selecting for, I don't know, the embryo with the lowest risk of developing type 1 diabetes, and at the same time, you are deselecting for another trait? It's of course very hard to know. However, all these predictors are validated in biobanks. Biobanks, remember, are these data of different countries with hundreds of thousands of individuals who are already probably dead. So we know whether they develop a phenotype or not, they have a disease or not. And all these data suggest that once you do simulations of the genetic selection, you are not really seeing the prevalence of an extra disease. And that's actually what opened the door to offer these predictors, because if we would have found like, oh, okay, in these patients with type 1 diabetes, you are actually, I don't know, going down in creativity, I don't know, then that will be a stop. But as of now, there is no data suggesting that. It could be true. However, as of now, we don't see that. We actually see positive pleiotropy, which is when you reduce the risk of type 1 diabetes, you reduce the risk of other disease because of comorbidity, which makes sense. But other traits, we still don't know.
Cool. All right. Well, thank you so much.
Oh, you have one question? Or?
Do you want to ask him? Oh.
Wondering, did you think of using the repair mechanism that exists in the cell? So the cell with the eggs, because the cell has a memory. Disease, it remembers how it looks. So we can use that mechanism.
Yes. So, again, CRISPR-Cas9 depends solely on not solely, but depends a lot on the cell repair mechanisms. But something that we found in this, we already knew in our field, but something we found in this project is that the human embryo is very inefficient in repair, because maybe it doesn't have that much memory there, right? When you create these gametes, they go through an epigenetic deletion or erasure. So we believe that's why it's so poor in repairing, and that's why we have aneuploidies. That's the most common genetic abnormality. Otherwise, we wouldn't see it. But that's the material we have with, and again, answering your question before, maybe we can inject something there in those embryos that will just enhance these mechanisms of repair, and hopefully that combination could be super efficient for this editing.
Amazing. Well, let's give our speaker a round of applause. If you have questions, he'll be around after, so feel free to ask more. Thank you guys for coming. Thank you. That was awesome. Hey, how's it going? Luke, Michael, here. Oh, perfect. Oh,
I was thinking